Vaccines are among the most successful medical interventions in human history. Smallpox, which killed an estimated 300 million people in the 20th century alone, was eradicated in 1980 through vaccination. Polio, which once paralyzed hundreds of thousands of children annually, has been eliminated from most of the world. The principle behind every vaccine ever made is the same: train the immune system to recognize a pathogen before that pathogen has a chance to cause disease, so that if exposure ever occurs, the response is fast and powerful enough to stop the infection before it becomes serious. To understand why this works, and why it works as reliably as it does, requires understanding how the adaptive immune system learns. When you receive a vaccine, you are introducing an antigen, a molecule the immune system can recognize as foreign, without introducing a dangerous live pathogen. Traditional vaccine antigens include killed or inactivated versions of the whole pathogen, live attenuated versions weakened by laboratory culture, or isolated protein subunits from the pathogen's surface. Regardless of the antigen type, the sequence of immune events is fundamentally similar. Antigen-presenting cells, primarily dendritic cells at the injection site, engulf the antigen, process it into small peptide fragments, and display those fragments on their surface using MHC molecules. The dendritic cells then migrate to nearby lymph nodes, where they present the antigen to naive B cells and T cells. A naive lymphocyte that has never encountered an antigen before and carries a receptor matching that antigen will bind to it, receive co-stimulatory signals, and begin a process of clonal expansion, dividing rapidly to produce millions of identical effector cells all targeting the same antigen. The key output of this process that gives vaccines their power is the generation of immunological memory. After the effector phase, when antibody-producing plasma cells and cytotoxic T cells are actively fighting a perceived threat, most of these short-lived effector cells die. But a subset, the memory B cells and memory T cells, persist for years or decades in a resting state. These memory cells have already undergone the clonal expansion and selection process. They carry high-affinity receptors for the antigen and are primed to respond much faster and more powerfully than a naive cell. On real exposure to the pathogen, the memory cells activate within hours rather than the days or weeks required for a primary response, and they produce a flood of antibodies and cytotoxic T cells before the pathogen has a chance to establish a serious infection. Memory is the biological substrate of immunity. mRNA vaccines represent a mechanistic innovation over traditional approaches. Instead of introducing the antigen protein itself, an mRNA vaccine delivers genetic instructions, a short strand of messenger RNA, encoding one specific viral protein, typically the spike protein of SARS-CoV-2. The mRNA is packaged in lipid nanoparticles that fuse with the membranes of cells at the injection site, releasing the mRNA inside. The cell's own ribosomes read the mRNA and translate it into the viral protein, which is then displayed on the cell surface or secreted. The immune system mounts a response against this displayed protein, generating antibodies and memory cells exactly as it would against a traditional antigen. The mRNA itself is degraded by normal cellular machinery within days. It never enters the cell nucleus and cannot interact with DNA. The idea that mRNA vaccines alter DNA is mechanistically impossible. mRNA is transcribed from DNA but does not reverse flow back into it under any normal biological process. Herd immunity, the protection conferred on unvaccinated individuals when a sufficient proportion of a population is immune, depends on the basic reproduction number, R0, of the pathogen. R0 represents the average number of people one infectious person infects in a completely susceptible population. The herd immunity threshold is approximately 1-1/R0. For measles, with an R0 of 12 to 18, herd immunity requires roughly 92 to 95% of the population to be immune. For polio, with a lower R0, the threshold is around 80 to 85%. Adjuvants, compounds like aluminum salts added to many vaccines, do not make the antigen stronger. They create a local inflammatory environment at the injection site that improves antigen uptake by dendritic cells and prolongs antigen persistence, enhancing the overall immune response. The vaccine schedule in infancy is designed around two biological windows: the waning of maternal antibodies passed through the placenta and breast milk, and the maturation of the infant's own adaptive immune system. Understanding how vaccines produce immunity also clarifies why they occasionally produce side effects like soreness at the injection site, fatigue, or mild fever. These are not signs that something has gone wrong, they are the expected footprint of an active immune response. The redness and swelling at the injection site reflect the innate immune response, vasodilation, increased permeability, and neutrophil recruitment. The systemic symptoms, fatigue, mild fever, and muscle aches, reflect the release of cytokines like interleukin-6 and tumor necrosis factor alpha that are part of the body's normal response to perceived infection. These signals actually indicate that the immune system is engaging with the antigen. The biological machinery activated by a vaccine and by a real infection is the same machinery. Vaccines simply allow that machinery to run its program in the absence of actual disease.